Looking closer at starch granules, we see again that they swell in water, and that this itself increases solution viscosity. These particles are larger than the wavelength of visible light and so make the solution opaque. As the particles actually dissolve, the starch molecules worm their way out into solution and make a much thicker sauce. Interestingly, we see that unbranched amylose can have a bigger solution profile than a branched amylopectin chain that has the same overall number of units. It's worth pointing out that these starch sauces can be broken down with too much stirring or handling. Common starch additives for foods are, well, besides flour or dried breadcrumbs, both of which work well enough most of the time, there's cornstarch. Again, there's corn, a New World species, or arrowroot, again, a New World species. So extracts of cornstarch and arrowroot are very useful thickeners. They're both simply starches that are purified and concentrated from their plant sources. So, reviewing, big granules of starch absorb water, thickening things, but the starch begins to leak out into the sauce. Eventually, the starch granules absorb so much water that they make a clear solution, but very viscous solution. Now, this slide we've seen before, and it's simply a reminder of the different kinds of starch found in a starch grain. Again, amylose, unbranched, amylopectin, branched. Plant glucans, or glucose polymers, come in branched and unbranched varieties, and again, they have different and useful qualities. In breads, for example, the branched amylopectin is useful because it's less inclined to lose its water or undergo retrogradation, that is to say, staling. Whereas for thickening, the unbranched amylose is superior because of its larger unfolded cross-section. So here's a starch granule, and this starch granule happens to contain a large amount of amylopectin, and you can see that these branched regions give rise to these crystalline layers, and these are where the water gets stored and is less likely, because of this crystalline structure, to be released and result in staling. Anyway, we're not talking about bread again today, so forgive me. Before we turn to proteins, however, we'll stick with starches for a few more minutes, or carbohydrates for a few more minutes. Let's think briefly on other non-glucose carbohydrate polymers that are useful in the kitchen as well. First, pectin, a key ingredient in jellies and jams. It's a sugar polymer derived from cell walls, so it's structural, not energy storage like starch. But note, too, that there are a couple of chemical details that are important to its kitchen properties. It's got this acid group right here that carries a negative charge at pH 7, but as pH is lowered, remember, lowering pH means hydrogen ions get more concentrated. The acid actually gets protonated and loses its charge. This effect is really wonderful as the negative charges at pH 7 make the chain repel itself and others, but at low pH, the likes repel problem goes away and the chains can associate to form, you guessed it, yet another open dynamic network of intermolecular interactions, the gel. In this case, it makes the solution seem almost solid. And indeed, this is the origin of the words gel and jelly, from the Latin jello, to freeze. The solution becomes seemingly solid, again, just like freezing water over. Now, many fruits are high in pectin concentration, but even with high pectin fruits, most cooks add pectin to help ensure proper texture formation. That is, those cooks that prefer more solid jellies. That is also a regional and cultural differences as well in jellies preferences. For myself, I noticed that the pectin content of the plums growing outside my kitchen actually decreases from June to July, so my later season preserves require more added pectin to avoid a soupy texture. As the fruit ripens, the pectin begins to break down. But let's look at some other carbohydrate polymers. These three groups of polymers are all derived from algae. That is to say, seaweed. I don't want to dwell too long here, but these are names that you will have seen on food labels. Of course, agar we all know from the familiar agar plates of the biology lab. The gel makes a wonderful solid substrate for growing microbial colonies on, and the carrageenans are a family of molecules purified from red algae in which the sugar monomers have been sulfated. There's a sulfur group for you. And finally, the alganates are particularly in vogue in fashionable restaurants because it's so easy to control their gelation. In this case, the molecule's acid group that's here is drawn as protonated. Please compare this with the deprotonated and charged acid group on this pectin structure. Remember, we're at pH 7, so this should actually show a negative charge. The alganates react through this negative charge with the positively charged calcium ions, and again, this charge neutralization means that the molecule is less self-repelling and forms a gel, much like the pectins do. And in terms of kitchen utility, they're tasteless, and they make a really good vehicle for delivering intense, designed, or defined flavor packets. Of course, these carbohydrate polymers are not just used to form gels, but are used as thickeners in diverse foods such as ice cream, candy, etc. So I hope you'll forgive me for excluding xanthan gum from this list and talking of interesting food additives. It's another complex carbohydrate polymer, this time derived not from algae but from bacterial cell walls, and we find it not only used as a thickener but as an emulsifier as well. Something else I don't have time to go into in this lecture is the interesting history of these polymers in foods. Suffice it to say that many of them have a long history, while others, like xanthan gum, are the products of modern product development processes. More again on this in later lectures. Okay, proteins at last, my favorite subject. But we're already quite familiar with the notion that in the kitchen, under stressful conditions of acidity and or heat, proteins lose their natural native structure. The same issue we saw in egg foams is also at play in sauce thickening. The protein of interest here is collagen, like here's a cartoon of the folded up form of collagen. Like the carbohydrate polymers in the previous slide, though, collagen is a structural molecule, oblong and rigid, and it lends this quality to the tissue where it's used, mostly connective tissues between muscles. Gelatin is not itself a protein, but is a substance or product that is composed of collagen. Structurally, collagen forms a triple helix, and you can see the three colors representing single helices. Recall the DNA double helix, but now we're talking about protein and a third strand for a triple helix. These three chains wind around each other normally, but at high temperatures, the denatured proteins become flexible and form these open networks that we're familiar with. As the solution cools down, the dynamic network becomes less dynamic and thickens the solution. And at still lower temperatures, the proteins form a much more rigid, but still very open structure. Again, a gel. I'm going to reiterate that the lecture is about sauces, and we're seeing the extreme here. However, we can use the same characteristics to thicken a sauce as well. This is a self-indulgent slide, so forgive me. Let's take a closer look at gelatins. Let's think about where they come from and their properties. As I mentioned, collagen is most densely concentrated in the cartilage and in the cartilage and connective tissue. Extraction for use as a gelatin then really requires processing bone and cartilage, mostly pigs, but I want to emphasize that those tissues are not commonly used in the home kitchen. There's enough gelling protein in the more delightful muscle tissues more than enough to accomplish the thickening required in most sauces. The other important thing about muscle tissue is that it's a much more concentrated source of the umami or savory flavor molecules, the free amino acids, especially glutamic acid. So there are other reasons for choosing a product to cook, aside from collagen content, obviously. Okay, now this again is going a bit farther than is really necessary for our discussion of sauce thickeners, but it's worth pointing out that fish is a fine source of collagen too. However, fish tends to be so delicate that other factors such as adverse flavor development might limit their use in the kitchen as thickeners. Of course, there may be cultural reasons for using fish collagen. Islam and Judaism both have restrictions on consumption of pork products, which again, pork being the principal source for most gelatins. But fewer restrictions on fish consumption. Likewise, many vegetarians do eat fish, so fish collagen might be an acceptable food additive for them. Okay, however, the seafood collagens also allow me to make a really interesting point that not all collagen is the same. There are subtle differences in the amino acid sequences and chemical cross-links between different species collagen that have real effects on the molecule's behavior. On the bottom left, we see that collagen derived from warm water fish, tilapia, form a more stable gelatin network than collagen from their relatives, the cold-adapted fish. So here we're showing the melting temperatures of these two fish, seeing that there's a 20-degree difference. This one, to the warm water fish, the gels melt at a much higher temperature than the cold-adapted fish. The figure on the right takes this a step farther and shows you something very complicated but interesting, but effectively the same idea as this slide, this table on the left. Let's see. You got on the y-axis here some spectroscopic signal that tells us about the protein. Let's just say we have a way of measuring whether the protein is folded up into its native shape or not. And when we increase temperature, note this x-axis is temperature, we see that the signal switches from high, whatever the signal is, high to low, denatured. And at the midpoint of the curve gives us some kind of diagnostic of the behavior of the collagen from these 5 different species. So this is collagen gelatin purified from 5 different fish species, and we see that each one has a different gelling profile. So it basically shows us the same thing. The sole is the warm water fish, and its collagen is more stable to unfolding by heat, which you might expect since it lives at a warmer temperature, than the cold adapted cod. And interesting, you see squid lying here somewhere in between, which is interesting. In any case, collagen is not just collagen, right? In any given organism, there are only over 20 different varieties of collagen, each of these with their own distinct properties. Perhaps an extreme example of this is an extreme example of the importance of knowing one's ingredients. But backing up to the gelatins in cooking, though, this is a picture of an aspic. The eggs and chicken are suspended or presented in a protein gel, and you can see that clear gel forming. I think it's fair to say that most Americans would not look favorably on this dish, not appetizing, but this kind of food object has certainly been popular during other eras in the United States and certainly in other parts of the world. More familiar to us, but no less a collagen-based product, is Jell-O. Many are the wonderful things that can be done with gelatin, and I just love the title of the book this image was taken from, Mrs. Beaton's Book of Household Management. Again, though, Jell-O had a heyday in the 60s and 70s and is more or less replaced in the fun diet of the modern child with commercially produced puddings, which, though they're still often collagen-based, have a smoother and creamier texture, less solid texture. Many homemade puddings are actually thickened with starch, and the most wonderful chocolate mousse is thickened by whipping air bubbles into the egg or cream sauce. But there are other food products that are very complex. A staple in my own household growing up was Cool Whip, which is actually an emulsion of hydrogenated vegetable oil and water, stabilized by milk caseins. I wonder why. I suppose people still eat Cool Whip, and I wonder why, though I find it really interesting that food products like Cool Whip are currently sort of frowned upon, while other more complicated products, thinking back to alginates and xanthans, are very popular in modern restaurant kitchens. Love the Jell-O brain. Interestingly, though, as an aside, I'll say that when we think of brain, we think of lipid, not collagen. So anyway, sorry, I always digress. Forgive me. Okay, so we were speaking of emulsions, however, when we were talking about Cool Whip. So let's go on to emulsions. We'll leave behind the tangly protein corner of the triangle and focus on mixing oil and water to thicken a sauce. In this slide, we see 3 different kinds of mixtures. On the left, oil droplets are suspended in a continuous phase, and that's a water phase. So you see continuous, I mean this is a continuous solution in which discontinuous droplets of oil are suspended. The emulsion is going to break down very rapidly, as you will see if you make a vinaigrette with no emulsifier. And in the second panel here, some kind of small molecule emulsifier, remember lecithin from egg yolks, provides a sort of a bridge between the oil and water phases and actually stabilizes that oil droplet in the dispersion. In the third case, we've added yet another layer of thickening, but including the open network of unfolded proteins to that emulsion, further obstructing the flow and stabilizing the emulsion. The recipe is worth noting. In an emulsion, there is a continuous phase, as I said, the phase that doesn't break down into droplets, and then there's a dispersed phase, the droplets themselves. It's the droplets that restrict the flow of the continuous phase. So the greater the proportion of dispersed phase, the more restricted the flow. Again, in mayonnaise, the proportion is some 80% dispersed oil to 15% continuous water, and the result is well known. Hardly a sauce at all, but a paste. However, mayonnaise is a paste that can be diluted to thicken a sauce in a very wonderful, delicious way. Okay, in this last slide, we see that emulsions can be created using either oil or water as the continuous phase. And it's worth pointing out that while vinaigrette might formally be considered a water and oil emulsion, that is to say, oil is a continuous phase, most recipes don't make this distinction and actually use oil as the dispersed phase instead, with no apparent or gross misfires in the product. McGee does point out that there might be a good reason for using oil as the continuous phase, and that's because it coats vegetables better. But the fact that so few casual cooks notice the distinction suggests that it might be really a meaningless distinction. So I've tried to make the case for you that there are several ways to thicken sauces, and I've tried to show you how each highlights a particular chemical or physical phenomenon, and that these phenomena are common to many other kitchen problems we've discussed. The items that are most glaringly missing from this discussion, spices and the dessert sauces like mousse, I've intentionally elided over. But fear not, we will discuss chocolate and foams and how they collide in desserts.